Hi guys, hello everyone. Hi, it's 12 p.m. already, so yes. So I have a lot of students who ask me why did I choose today to do live instead of next Thursday. It's because that the reason why I chose today, just in case you all really like don't remember anything for biology, your brain only got like any other subject besides biology, so I decided to do it today. And the other reason is because the video can be replayed if you didn't get to do it today, so you can also replay the video maybe next week or after you finish your, I think it's at next paper. So you can actually do that lah, okay? It can be replayed. So yeah, and I also noticed that I clash with a lot of tutor times today because I know that you have NX and you have chemistry. So I actually clash with my own chemistry tutor timing at 12 o'clock. But never mind lah, my video can be replayed lah. So yes, by the way, the file I have attached at my Google Drive, so you can download the file at Google Drive. So same thing, we have two file, two file. One I actually type it, the other one is the original English, the BM and English version for Mrsm. So if you are taking the BM paper, like BM version of biology, you can refer to the original file. If you are taking only English, you can refer to my Microsoft Word, which is the PDF file that I type it because I actually align it into a better arrangement. So it's your choice now, okay? It's your choice to do that. Hi guys, for those who just came in, yes, hopefully that your brain still got some biology information right now. Hopefully it's just not just like chemistry and admax only. I think next week is only admax and chemistry. So yeah. Okay, so for another 1 more minute, we're gonna start. While waiting, you can just like print whatever stuff. I hope that you have already print your stuff already. Hopefully. Yes, right. Hopefully that you have print your stuff. Wait, Andrew! Hi, Andrew. Why you here, Andrew? I was very surprised that you are here, Andrew. I thought you will go to chemistry class today. Like now, UTN chemistry class. Because, like I thought UTN chemistry class end at 1:30. So I'm a bit surprised that you are here. So yeah. Okay, so I think we can start now. But I want to say that I think the class today it will be slightly longer. There are a lot of things that I want to explain, so it will be a very long hour class. So you can prepare whatever thing that you want to prepare first, your water bottle and your food lah. Hope that I will not rap today because actually for the previous SBP video, right, I didn't really rap though. I mean, if you guys notice, I just used a very slow speed. So today maybe I will speed up a little bit. So if you are thinking I'm too fast, you can always pull the thing back and you can watch it again. Okay. So MRSM paper 2. This paper is a paper that you guys chose the most in my Instagram voting. The paper here is not to say very hard, but some of the questions, some of the subtopic are quite tricky. Okay, it will not be hard, but it's tricky. Okay, so we're gonna start the class. Hi, Sean. Okay, let's do the first question. So as I mentioned previously, right, the first question it will be Form 4 Chapter 2 or Form 4 Chapter 3, right? So based on this question, you can see it's Form 4 Chapter 3. It says 1.1 shows the structure of plasma membrane, which is known as fluid mosaic model. Name protein X and protein Y. This is a very easy question. For every question that you have to know in biology is the labeling first. If you don't know the labeling, there are a lot of questions that you cannot do, can do. So I have mentioned 1,000 times. You must understand and you must know the labeling. So first of all, how do I want to differentiate whether it is a pore protein or whether it is a carrier protein? Very simple. Pore protein is always open. Can you see here? This one is always open. So this is a pore protein. Next one, we have protein X. Protein X is closing of one part, right? So if one part is closing, we call it as a carrier protein. All of the carrier protein, they have their own active site. We call it as binding site. And we also label structure Z as well, okay? Structure Z is 2 layer. This 2 layer here, we call it as phospholipid bilayer, okay? What do you have to know besides the name? You have to know what are the structure that can pass through these 3 areas. So first of all, we're going to start from pore protein first. Pore protein is a protein that can carry out 2 processes. First, they can carry out osmosis. Second, they can carry out simple diffuse, not simple diffusion, facilitated diffusion, okay? So they can carry out two processes. So if it's osmosis, right, the only example they can transport, it will be water molecule if it's osmosis. But for facilitated diffusion, the pore protein, they can transport a charged ion, okay? Charged ion such as potassium, sodium, those are known as a charged ion. If it's a carrier protein can only be used in facilitated diffusion. Facilitated diffusion, the example over here, it can only be a large molecule. Large molecule. So what are the example of large molecule? We will have glucose and amino acid. Those are large molecule. Phospholipid bilayer, they will carry out through two processes. The first one is osmosis. The next one is simple diffusion. So osmosis, of course, the example can only be water. If it's simple diffusion, we have more example. We have like lipid soluble substances. We have dissolved gases. And we have the last one, which is small molecule. You have to know the example of each of the thing here. So for lipid soluble substances, right, we have vitamin A, D, E, K, fatty acid, and glycerol. If it's dissolved gases, we have oxygen, carbon dioxide. Small molecule, we have water. So you have known all of the example here, just in case they ask you to write example, or they ask you to write the process that it can carry out by the carrier protein, pore protein, or phospholipid bilayer. So we have done with the first one. They ask you what is the characteristic of Z. Z is a phospholipid bilayer. We did it in SPP paper, right? So for phospholipid bilayer, you have two parts, the head and the tail, right? So the head, we call it as what head? The head is the one that face outwards, right? So it's the one that face outward. It is known as a hydrophilic head. Hydrophilic means they attract to water. We call it as hydrophilic. The tail face inwards, we call it as hydrophobic. So you can just write that the head and tail, one is attracted to water, one is repel against water. Just for active transport, it uses carrier protein, right? Correct. For all of the active transport, they will use carrier protein. Because this one is down the concentration gradient, so I just use passive transport. So this one you can write. They have hydrophilic head, which they attract to water, and hydrophobic tail that repel against water. Okay, so this is the thing that you have to write. Please make sure that you can form it into sentence lah. Okay, just write hydrophilic head, which is attracted to water. Hydrophobic tail, which is repelled against water. Just form your sentence lah. Okay. Next one, the following substances move across the plasma membrane through passive transport. So it means that this entire thing is a passive transport. They give you example. You have to fill in according to the structure. Z is phospholipid bilayer. X is a carrier protein. Y is a pore protein, right? So first one, we're going to start from Z. Z is a phospholipid bilayer. Just now we mentioned, right? Phospholipid bilayer, we have few characteristics that can pass through, such as lipid soluble, such as dissolved gases, and also one of the example, which is small molecule. So over here, which one here is under the three category? Lipid soluble, small molecule, and dissolved gases. You have only one answer, right, which is vitamin E, because vitamin E is lipid soluble vitamin. So the answer for Z is vitamin E. Next one, for protein X. X is a carrier protein. So this carrier protein, right, in this question here, they can only transport large molecule. So as I mentioned just now, large molecule, we have amino acid and glucose, right? So the answer here is amino acid. And then for protein Y is a pore protein. Pore protein in passive transport, they transport charged ion. So charged ion here is potassium ion, and you only have one choice. So it's charged potassium ion. Hi, Julia. Well, you really come, Julia. Okay, next one. The movement processes of substance in 2B1 is categorized as passive transport. Explain why. How do we want to explain what is a passive transport? So we all understand we have two transport. One is active, one is passive, right? So for active transport is the one that requires energy in the form of ATP. They will move against concentration gradient. That is active transport. Passive transport is the other way around, where it does not require energy. It moves down the concentration gradient. So if you cannot differentiate what is active, what is passive, you can make it into a simple word to understand it. Active is you do it by yourself. Passive, someone do it for you. So for example, passively, someone do it for you, right? You don't need to use any energy. And going downhill is easier compared to going uphill, right? So passive transport, it moves down the concentration gradient, and then for active transport, it moves against. So for this one, only two things you have here, right? The concentration gradient and does not require energy. So there are only two things. So you can say that it does not require energy. Substances move down the concentration gradient. Okay, if you don't want to use the word down the concentration gradient, what are the words that you can replace? You can replace with high to low concentration gradient, high to low, or you can replace with follow the concentration gradient, or along the concentration gradient, whichever that you prefer also can. So these are the two things that you have to write for passive transport. So if the question asks you for active, I think you all should know the answer for active as well. I just opposite meaning, okay? Okay, so I think the question that student always gets stunned for MRSM paper 2, right, is the following question, which is this one. They give you a COVID-19. And whenever people see COVID-19, right, they will get panic. Confirm they get panic, okay? It says 1.2a shows the structure of a novel coronavirus, COVID-19 virus. 1.2b shows the molecule of structure of the soap. So 1.3 shows the action of the soap molecule on COVID-19. So molecule has a similar structure as phospholipid layer on plasma membrane. So the question says that 1.2a, this is the virus, okay? For 1.2b, this is the soap, right? So it says that the soap has a similar structure as a phospholipid bilayer. So just now we learned that phospholipid bilayer, it has two things, right? Which is the hydrophilic head and hydrophobic tail, right? Guys, don't worry. Whenever you look at this question, right, you think about it. What chapter is this? This is Form 4 Chapter 3, right? So it's something to do with your Form 4 Chapter 3, like the plasma membrane. Of course, you have to understand what are the chapters is about. Only you can try to link the thing together. So this question is very simple. Okay, now, based on this question, right, so we have hydrophilic head and hydrophobic tail. Let's talk about the characteristic of the hydrophilic and hydrophobic. So just now we mentioned, right, hydrophilic head is the one that they attracts to water, right? Hydrophobic tail is the one that repels against water. What does the question say? The question asks you to do what? Based on 1.3, how the practice of washing hands with soap and water can destroy the virus. Very simple. You based on your normal common sense plus a little bit of your biology knowledge, you can answer this question. Okay, so you have water and you have soap, right? Soap is the one with hydrophilic and hydrophobic, and water is H2O. So based on these two characteristics, which is hydrophilic and hydrophobic tail, right, we will understand that actually the hydrophilic head, they will react with water, right? Because hydrophilic means they like water. If they like water, of course, they will react with water, okay? If it's hydrophobic tail, they don't like water, right? So what they can react? They can only react with lipid-soluble substances. So this one, they will react with the lipid. Where is the lipid come from? The lipid come from the virus. Can you see here is what? The lipid membrane and other proteins on the virus, right? So this one is the virus they are referring to. Oh no. Wait, these are the virus they are referring to, okay? Now, over here, what you can see here is what? You can see there's a diagram over here, right? It's like water to wash your hand again. It's like to wash away the virus. So whenever you wash your hand, it's supposed to be like 2 minutes according to the Ministry of Health, okay? So you have to like, wash properly in order for you to kill the virus, because this is how it actually works on the virus. So you based on what you learn in your biology and you learn in your no sound. Is there no sound? Got sound? No sound? No sound? Got sound? Because I don't know whether got sound or no sound. Can anyone tell me whether I have sound or not? Or is only the physics tutor got no sound? Okay, got sound. Walao eh, Wei En cannot lah you. Aiyo, scared me way. Okay, so back to the question. Okay, we're gonna form sentence from here, okay? Now my iPad lag a little bit again. Okay, let's form sentence based on what we understand just now. Hydrophilic and hydrophobic, okay? Got sound lah, yeah, scared me. Now my iPad lag. Oh my goodness. Okay, give me like 30 seconds, okay? I need to share my screen again because it's lagging. Stop sharing. Share screen. Wait, give me about 30 seconds to share my screen. Okay, so that it won't lag. So I can write better. Just now suddenly got no sound for a while. Okay. Can. Thanks. No, I just want to make sure that I can write better today. Unlike last time. Unlike last time, okay? The MRI, the SPP paper, like suddenly I cannot write anymore, right? So this one, I know the trick to make sure I can write again is to share my screen again. Okay, so form into sentence, you can see hydrophilic head of soap molecule will bond with water. Hydrophobic tail of soap molecule will bond with the lipid membrane on virus, where the soap destroy the membrane of virus. Okay, if you want to explain, because the question is only 2 marks, right? You can actually write more if you want to explain, but this 2 is enough for you to get the 2 marks. So if you want to write more, then you can write, this causes virus to break apart and soap trap dirt and the micelle. Micelle is the tiny bubble that contains of the destroyed virus. Based on the question they give you, right? Micelle, it is a bubble that contains of the virus. Trap dirt and micelle, which then wash away in water. Okay? Just in case you guys understand this one, this is dirt. So if you want to write the first 2 sentences, which is totally fine because it's only 2 marks. So guys, whenever you see this kind of question, right, you base on what you understand in your biology. And based on the question, because they actually give you the hydrophilic and hydrophobic, right? So you based on the 2 points, you can explain on your answer. Okay, so you have to know what is the keyword in the sentence. So the keywords over here is the soap and the water. So soap is the one that has the ability to bind with the lipid, because lipid has, the virus has a lipid, right? So you have to understand the keyword in the question. So it's not to say very hard, okay? Next one, 1.4 shows a package of an oral rehydration salt. I forgot to find a better diagram. I was thinking to find a better diagram, but never mind, it's not important, okay? Oral rehydration salt. Okay, oral rehydration salt, it is a isotonic drink. For example, people who has diarrhea, it will drink ORS. People who carry out vigorous activity, they will also drink ORS. It says individuals who suffer from diarrhea is advised to drink rehydration salt such as oral rehydration salt to replace water loss in the body. Explain why. In ORS, it contains of 3 things. Number 1, it contains of water. Number 2, it contains of electrolytes. Electrolytes means salt, like for example, sodium, potassium, those are known as electrolytes. Because as you know, when a person has diarrhea or when a person carry out exercises, right, you will actually know that you will sweat a lot. Those sweat contains of charged ion, like electrolytes, okay? So the third one, it will be glucose. Why do you need glucose in rehydration salt? It's because that you need to generate more energy. So that's why these are the thing can be found in ORS. For example, 100 plus, it can be one of the example of ORS because 100 plus is easier to get compared to the ORS, because ORS is a package that you have to add with water one. Apparently 100 plus itself also come out with ORS lah. So yeah, I'm saying the 100 plus carbonated drink lah. That one is easier to get in the supermarket or even in KK Mart. But ORS, you have to get it like pharmacies, or you have to get it in a specific area, only you can get ORS. But it's cheap though, like one package is like 1 ringgit to 2 ringgit like that. So for those who have diarrhea, you can try this instead of charcoal peel also can. Both also can eat together lah. So you have to explain why do you think they have to drink ORS? Because people who have diarrhea, they will lose a lot of water and mineral as well. So you can see diarrhea patient lose a lot of water that causes an increase in blood osmotic pressure. Okay, what is a blood osmotic pressure? For those who still don't know what is blood osmotic pressure, the easiest way is to see how much of solute in your bloodstream. How much of solute? Whatever that is not water, we call it as solute. So for example, when the question says high blood osmotic pressure, it means that the body has a lot of solute. Like people who carry out vigorous exercises, people who have kidney failure, they will have a lot of solid in the body, right? We call it as high blood osmotic pressure. If it's lower blood osmotic pressure, it means they have lesser solute. Lesser solute. Lesser solute means they have more water. So as a normal human, we're supposed to have a balance point between blood osmotic pressure. So since this person has a higher blood osmotic pressure, it means the person's body has lesser water. It's called high blood osmotic pressure. Lesser water, more solute. So you have to continue to explain where I can say that the rehydration salt contains of salt, sugar, and water, which able to replenish water and electrolyte loss. So if you want to explain about the sugar, you can say that the sugars will help in generating energy. That is the answer. Okay, now done with this one, we're going to move towards to question 2. Question 2 is Form 4, Chapter 4 under, this is carbohydrates. So carbohydrate compared to protein, lipid, and nucleic acid, actually, there are more things to learn and there are more things to remember as well. So the question gives you 3 diagrams here. Honey, they give you a vegetable and milk. You have to categorize what are the example of these things. Honey, okay, by the way, just in case you all don't know, okay, we have 3 different types of sugar. Mono, di, and polysaccharide. You have to know all of the example over here. So for monosaccharides, right, there are 3 examples, 3 for each. We have glucose, fructose, galactose. And then for disaccharides, they have lactose, maltose, sucrose. Polysaccharides, they have glycogen, starch, and cellulose. These are the examples. So you have to know the example is for what type of fruit and vegetable. So the first one is honey, right? So honey, it is actually under glucose or fructose, okay? So they're actually under monosaccharides in this case. Because sucrose can be broken down, right? So fructose is a fruit, and then for glucose is also from fruit. So this is actually monosaccharides for honey. This is monosaccharide. Okay, because whenever you name the type of carbohydrate, right, you don't name the example, you name the type of carbohydrate. You don't name the example. Then for food, Q, right? Q, they are under vegetable. So vegetable, right, you can only think of the last one, which is polysaccharides. Why? Because polysaccharide, they have starch and cellulose, which is the characteristic that vegetable have, right? Vegetable, they have cell wall, they're made of cellulose, and they will store the glucose as a starch. So the answer is polysaccharide. This one is polysaccharide. And it says 2.2 shows the part of the word equation for breaking down of food sample R. So food sample R is milk, right? Milk can be disaccharide, it can be monosaccharide. But obviously that this diagram here, food sample R is a disaccharide. Why? Because they give you lactose. When you break down lactose, process M is what process? Breaking down process, it is known as a hydrolysis. What does hydrolysis mean? Lysis, it means breakdown. Hydro, it means water. When you break down, it requires water molecule. We call it as hydrolysis. When you break down lactose, because lactose is disaccharide, right? Di means 2. It must be 2 equals to 1 plus 1, right? So the answer must be 1 plus 1. What is the 1 plus 1? The 1 plus 1 is the monosaccharide. One of the example here, it must be glucose, okay? Must be glucose, okay? Must be glucose. The other one, and because this is milk, right? As I mentioned, right, milk can be lactose. The other example, it can be galactose. So it's glucose plus galactose. Glucose plus galactose. That is the answer for X and Y. But the question did not ask you to write what is X and Y. They asked you to explain process M. So if you don't know what is X and Y, actually still okay. Okay? Still okay. For sentences format, you can just write it produces X and Y. But if you know the example, it will be the best, which is glucose and galactose. The question is 3 marks. The first one, we have to talk about what is process M. So you can say that process M is hydrolysis. So lactose is broken down using water, glucose, and galactose is produced. Actually, for every condensation, they will have a bond between them. Like for example, this carbohydrate, all of the carbohydrate here, it comes in a hexagon shape, which is like that. Hexagon shape. Okay, so if it's a disaccharide, this is how a disaccharide looks like. Because when you break down, you have to add water, right? Then it will break down into mono plus mono. So there is no bond between mono and mono. You can see there is a line between over here, right? So this line, actually, it has a name. But it's okay that you don't know. Just in case you need to write the bond name, the bond name is known as a glycosidic bond. Okay, it is known as the glycosidic bond. Just like protein and amino acid and amino acid, right? Together, they have a bond between them. They call it as a peptide bond. So for all of the breaking down process, right, they requires enzyme because enzyme is the catalyst. So if you want to write enzymes over here, it's your choice. But if you don't want, totally fine as well. If you want to break down lactose, the enzyme that you need, it will be lactase because every enzyme is supposed to end with ASE. If, for example, protein enzyme don't end with ASE. Protein enzymes ends with IN. For example, you have erepsin, pepsin, and trypsin. Those are the protein enzyme. So all of the carbohydrate enzyme and also lipid enzyme, they ends with ASE, okay? And they ask you to write the complete word equation represented breaking down of food sample R. Food sample R is the lactose, right? Continue. So right now at this question, you have to know what is X and Y. For this question, you can just write X and Y is produced, which is fine, which is also correct. But at the next question, you have to know what is X and Y since we know the answer, right? So word equation means what? It must be word. Okay, you cannot write what H2O. You cannot write CO2. Okay, no, must be word. So basically, it's lactose plus water. It breaks down into glucose and galactose. Okay, so for example, let's say they give you this diagram, the one that I draw over here, right? How do I know which one is glucose, which one is galactose? Actually, both also correct. You can write the front one as galactose. You can write the back one as galactose also can. As long as your products names are correct, your answers are correct. So the arrangement doesn't really matter lah. You want to write glucose first also can. You want to write galactose first also can. So you must know the rest of the disaccharides formation as well. For example, you have to know sucrose, you have to know maltose as well, just in case they give you the other example. Okay? Next one. 2.3 shows the structure of an enzyme and 3 possible WXY substrate. And they ask you to choose the correct substrate and complete the schematic diagram below to show the action of enzyme in food sample R. So how do you want to choose a proper one? This thing we call it as lock and key hypothesis. What is a lock and key hypothesis? Enzyme must act as a lock, and substrate will act as a key. Only the correct key can open the correct door. Like for example, my house key and your house key. Can we open each other door? Of course cannot, right? Because the shape is different. If can, right, then very dangerous already, you know. So you have to find the specific shape because all of the active site over here, all of the enzyme, they have this part here called active site. So they must have a specific shape. So obviously in this diagram, the one that fit into enzyme, right, it will be Y. Because this part here, it fits. This part here is fit, right? So the answer is Y. Now we're going to draw the diagram. So first we're going to draw Y, right? Okay, my drawing very bad. If I draw very ugly, right, I mean, you must draw better, okay? Because I don't want to spend time like drawing a very nice diagram. Okay, so this is the substrate Y. And then you have to form a next thing. Next thing we call it as enzyme substrate complex. This enzyme substrate complex is very easy to draw because they already give you a shape, right? You just draw slightly outer layer, like slightly outwards, and then you draw a straight line downwards and like that. Ta-da! This is the diagram. That's all only. The last one, we will have the products. So how do you want to draw products? Imagine this thing is being cut into half. So if you draw the upper part and the bottom part, right? So we have the upper part like that. Yeah, a bit ugly, but never mind, okay? And the bottom part. Okay, so this is the product. Doesn't matter which one is glucose, doesn't matter which one is galactose. Doesn't really matter. If they ask you to label, it doesn't really matter, okay? Now, drawing itself is a very easy question. Asking you to find the correct key also very easy. The next question is a test question. This one, actually, you learn it since like Form 2 until right now. It says 2.4 shows the sequence of color changes in Benedict solution when 2 ml of this solution is added into 5 ml of mango juice and it is heated in water bath at 37 degrees Celsius. So 37 degrees Celsius, it is the optimum temperature. It says the color changes, right, is from blue to green to yellow to brick red precipitate. This one here shows what? It shows that mango juice, it is a reducing sugar. What is a reducing sugar? Reducing sugar, it means they are able to convert copper 2 ions to copper 1 ion. Where does the copper 2 ion come from? Benedict solution is copper 2 sulfate. Copper 2 sulfate. So copper 2 sulfate, the copper 2 ion, it is blue in color. Hopefully that you guys know copper 2 is blue in color. If it's not, then your chemistry teacher will be very sad also, okay? Copper 2 ion is blue in color. And when it changed to brick red precipitate, so basically it's from copper 2 to copper 1. That's why it is known as reducing sugar. What are the examples of reducing sugar? All mono and di, besides sucrose. What are the mono and di? We have glucose, fructose, galactose, lactose, and maltose. It is known as the reducing sugar. If you add everything here, not everything, like one by one, okay, the result will be the same, which is a brick red precipitate. And then we have to explain the result, right? Because copper 2 is blue, copper 1 is red. So when you add mango juice, which is a reducing sugar, it will change the blue solution into brick red precipitate. So you can say that mango juice contains of reducing sugar. It can be any example, okay? If you do want to write example, also can. Next one. You can say that it reduce copper 2 ion, which is blue in color, to copper 1 ion, which is red in color. So this is the answer for copper 2 to copper 1. But as I mentioned, if you want to write the example, mango juice contains of monosaccharides also can, glucose, fructose also can, but it's not lactose obviously because it's not milk, right? So it's not lactose, it's not galactose. So you can write glucose, you can write sucrose, glucose, maltose, and fructose. It will not be galactose and lactose because galactose and lactose is for milk. Okay, next one, we have 2.5 shows a quarter, half campaign introduced by the minister, this one Cancer Minister of Health, to promote healthy eating habits among Malaysians. Explain why we should take only quarter portion of rice in our daily diet. So why do we need only take quarter portion of rice? It's because that rice itself, it is the basic unit is glucose, right? Because they are starch. So the basic unit is actually glucose. When you eat a lot of glucose, right, if you cannot convert into a glycogen, they will store as a fat. So the person might have increasing of your blood sugar level. It will lead to diabetes mellitus, or they will lead to obesity. So that's why for Malaysian, right, we have a lot of people with diabetes mellitus and obesity, or even high blood pressure problem, because that we don't really lack of food in Malaysia though. And for the homeless people, they are being fed very well by the NGO in like KL or whatever lah. Okay, so that's why you only have to eat quarter portion, especially kids this day. They eat a lot of food. Okay, so if you're thinking that you're obese, right, please reduce intake of rice. I'm not saying that you cannot eat rice. I'm just saying that reduce intake of rice. Especially, I have a lot of students, girl students, they always ask me, how do you want to reduce your body weight? Just eat lesser rice. That's all. Or you can just stop eating rice, eat a lot of protein, and also eat a lot of vegetable. Okay, so you can see that this can reduce risk of diabetes mellitus, because rice contains of starch that will produce large amount of glucose. So this is actually the answer. Okay, next one we have question 3. Question 3 is under cell division, mitosis and meiosis question. For those who still don't know how to differentiate mitosis and meiosis, don't worry, I have a video, so you can watch back the video about how to differentiate mitosis and meiosis. So it's your choice to look at the video lah, okay, if you still don't know how to differentiate. So by looking at this diagram, you can see you have cell X and cell Y. So cell X and cell Y, how do we want to differentiate? For meiosis, we have to know one fact is, we have to know that meiosis, they have 2 different colors in one single chromosome. So you can see, this one is 2 different color, correct? 2 different color in one single chromosome, right? So this is obviously is meiosis. And you can see this one single color in one chromosome. This is mitosis. So now that we know that this is mitosis and meiosis, right, we have to know the stages of it. For cell X, they are in the middle. Since they are in the middle, it is known as a metaphase. Metaphase. And then for meiosis, right, they are being separated and they are still in the butterfly shape. Like chromosome, as a complete chromosome is a butterfly shape. So it's in the butterfly shape. This is a anaphase 1. Because anaphase 2, right, is half, which is sister chromatid. They ask you what type of cell is involved. Type of cell involved, right, it will be whether animal cell or plant cell. Because for the type of cell, in this diagram here, it is round in shape. Round in shape, there is no cell wall. Number 2, if you notice, it has a centriole. Only animal cell will have centrioles. Plant cell has no centriole. So obviously that this is animal cell. And they ask you to explain your answer, right? This one, number 1, no cell wall. Number 2, it has centrioles. Plant cell don't have centrioles. Next, name the stages of nuclear division shown by cell X and cell Y. So we have written the answer on top. So X is metaphase, Y is anaphase 1. Okay, anaphase 1. You must write 1. Cell X in diagram 3.1 divides 4 times. State the number of daughter cell produced. The keyword here is what? Daughter cell. Daughter cell means that one cell, it will form into 2 cells. And these 2 cells we call it as daughter cell. So in this case, if they divide 4 times, if you are not good with calculation, you don't use 2 times 4, guys. If you don't know, you draw things out, okay? Like 1, 2, 3, 4. But there's a lot to draw lah. Let's say what if the question says divide like 5, 6, 7, 8, 9 times. Then you cannot draw everything out, right? So the easiest way, you use 2 power of 4. Okay, the 4 times is the 4 times. If 5 times, then 2 power of 5. You use power of, you don't use times. So 2 power of 4, the answer is 16. But if you don't are not very good at this one, you just draw things out, you just draw everything out. Draw 4 times, then you have your answer lah. But if it's 10 times, you cannot draw, right? Then you use 2 power of 10 calculator lah, okay? Or you use your count. State the number of chromosome in each daughter cell. Okay, number of chromosome, right, in the daughter cell. Okay, started number 1. It started with 1, 2, 3, 4, 5, 6. 6, right? It started with 6, right? So the parent cell got 6. And for cell X, it is a mitosis. Mitosis, it will never change the number of cells. So if it's 6, the daughter cell will also be 6 because they want to maintain the chromosome number in a cell. Like for example, it will never be 3 because if it's 3, right, that one is a reproductive cell already. So the answer must be 6. It follows the parent cell. If it's half, only will happen in the reproductive organs, which is the sperm and ovum. Those are half. Next one, question. In the box provided, draw the possible daughter cell for Y at the end of cell division. Cell Y is meiosis, right? At the end of cell division, you should draw which phase? Should you draw telophase 1 or telophase 2? End of cell division, you must draw telophase 2. Okay, must be telophase 2. Confirm must be telophase 2. First, you draw a circle. Okay, first circle. And then in the diagram, right, we have to count the number of chromosomes first. In this diagram, how many here? We have 1, 2, 3, 4, right? So when they separate into 2, because this is anaphase 1, so they become telophase 1, right? They will have 2 here and 2 here, right? And it will be separated again. So it becomes like half half. So whenever you draw, you can only draw half half, which is like that. 1, 2. Okay, 2. But do you think drawing like this is complete? No. If it's meiosis, it must be different color. It can be full color white or full color black. It's your choice. But usually I will just draw half color lah, okay? Like this is half darker color. One of it got different color, your answer is correct already. It doesn't matter what is the combination, you just draw this one, your answer is correct. As long as you have different color in one single chromosome. Okay, next, we're going to look at the next question. 3.2 shows another cell Z in one phase of cell division. They say chromosome P is not separated. Complete the diagram of 2 daughter cells. They will be formed in the space provided below. In this diagram here, it says P cannot be separated. Okay, let me draw this entire thing as red color. Okay, supposedly during anaphase, right, it's supposed to be pulled apart, right? So this entire thing goes to this direction. This part here, it will pull towards this direction. This part here, it will pull towards this direction. This one here, it will pull towards this direction. And then the leftover part, I use purple color lah, okay? This other part, it will move it to opposite direction. So whenever you draw the diagram, it's very simple. Red one in one circle, purple one in one circle. That is your answer. But when you draw, you have to make sure that the size of chromosome, you must follow the diagram. So the first one, you have this diagram, right? The bigger one is in darker color, the one that is not separated. Yeah, I don't want to color so much. You all can color better, okay? And then you have the smaller one, which is white in color, a bit too big, smaller one. And then you have the longer one, which is white color, and the smaller one is black color. So you must follow the size and the color of chromosome. And for daughter cell too, right? You have a smaller black color one, and then a smaller white color one, and a bigger white color one. The arrangement doesn't matter as long as you have the result like that. Your answer is correct. Okay, you all color properly and draw properly lah. Okay, I don't want to spend time drawing the diagram. As long as you have the same diagram here, your answer is correct. Okay, so for next question it says 3.3 shows the condition of tumor cell. Statement 1 describe about the tumor protein in P53 in cell cycle. Based on diagram 3.3 and statement 1, explain how the substance in cigarette smoke leads to tumor growth. In the statement here, it says that tumor protein 53 is a gene that regulates cell cycle and helps to prevent development of tumor that suppress cancer. If this thing cannot work, it means that the cell will carry out uncontrolled mitosis. Why? Because it says that explain how substance in cigarette smoke. Cigarette smoke contains of carcinogen. Carcinogen is the one that leads to cancer, right? So it will stop the protein 53, P53 from regulating the cell cycle. So you cannot control how much of cell that you want to produce. So when you produce too much of cell, it will lead to cancer cell or cancer cell, it will lead to tumor cell. So that's why whenever you see something new, don't worry, go back to your basic only. Anything, just go back to your basic, okay? So for this one, you can see that cigarette contains of carcinogenic substances. This stops the function of protein P53 and cell undergo uncontrolled mitosis, which causes formation of abnormal number of cells. Okay, so this is the answer. Don't have to worry about whether you can do or not. Of course you can do. Just go back to your basic. So don't always think that K-bar question is very, very, very hard. Sometimes it's just the basic that you have to know lah, okay? Just go back to basic, okay? Unless your basic is very bad lah, then you have to like practice on your basic lah, okay? You have time to practice on your basic, okay? Next one. Question 4. 4.1 shows the female reproductive system. This is under 4.5 chapter 4. You have to know the labeling and you have to know the process of female reproductive system. It says, name structure X. Obviously the X, this part here is the ovary. Very easy, right? Ovary. This one you don't know, I'm a bit scared. Okay, ovary. State one function of the hormone secreted by X. Ovary has two different hormones. The first one, they have progesterone. The other one, they have estrogen. So they have two. So they have two different hormones. Because for female menstrual cycle, we have four hormones, right? Which is FSH, LH, progesterone, and estrogen. The one that is not in short form, obviously that one is from the ovary. It says ectopic pregnancy is the condition where fertilized egg does not implant to the uterus wall. Based on the above statement, label the part where ectopic pregnancy may occur by using letter Y on diagram 4.1. So for this question here, right, ectopic pregnancy it does not happen in the uterus because they say implantation does not happen in the uterus, right? Usually for ectopic pregnancy happens in the fallopian tube. So this is why usually lah, because rare cases they will implant in the cervix. Okay, rare cases, but usually it's the fallopian tube. Why? Because fertilization takes place at the fallopian tube, right? When the zygote cannot move downwards, it means that they cannot go for implantation. So what will happen to the mother? Eventually, the mother has to abort the baby. Either the mother abort the baby or the mother will die due to excessive blood loss because the baby will grow bigger in size, right? Until one point, the fallopian tube it will burst because fallopian tube is not elastic. Uterus is elastic. Only uterus can expand. Fallopian tube cannot expand, so it will burst. Anyhow, the baby will die lah because you cannot save the baby. Teacher, you eat lunch already or not? What do you think? It's 12:47 now. Okay, so next 4.2 shows the development of follicles and the thickness of endometrium wall menstrual cycle. Based on 4.2, name the phase S and T during menstrual cycle. So phase S and phase T. Phase S is actually the phase where you have bleeding. We don't call it period, guys. In biology, we call it as menstruation. Okay, menstruation. So for S, we call it as menstruation. Menstruation. And then for T, right, you can see there is thickening of endometrium wall. There's a lot of blood vessel inside, right? It has a phase, they call it as a vascularization. This is R. Vascularization. It's known as a thickening of your endometrium wall, which contains a lot of blood vessels. So that's why whenever a female undergo menstruation, there's a lot of blood coming out because of this process, vascularization. And you have to draw the diagram, right? If it's not mistaken, they ask you in the space provided 4.2, draw based on the following information if fertilization occurs. Okay, the keyword here is what? The keyword here is fertilization occurs. It means the person is pregnant. When the person is pregnant, they must have high level of progesterone and estrogen. Where does the progesterone come from? The progesterone comes from the corpus luteum, which is this one, corpus luteum. Since there is fertilization, means the person is pregnant, the diagram here, it will be the same diagram. Okay, they will maintain up to 3 months lah, okay? And then 3 months later, it will take place by the placenta. So the corpus luteum is to maintain the production of progesterone, to maintain the thickness of endometrium. So the endometrium wall, it will remain high. Okay, it will remain high. So this is the diagram. So that's why for pregnant women, it's important for them to have progesterone and estrogen, because if they have lesser, right, eventually it will lead to miscarriage. So that's why they have high. So if you watch drama, then you should understand, no, when people fall down, there's blood coming out, it's because the endometrium wall breakdown. Then you have like, the baby is gone lah, based on the drama that you all see lah. It's something like that lah. Next one, explain how contraceptive pills prevent the process N from occurring. Okay, you have to look at what is process N. Process N is on day 13, right? Day 13 to day 14, right? So you can see that they will release secondary oocytes. Releasing of secondary oocytes, it is known as ovulation. So process N is ovulation. Ovulation. So contraceptive pill, it contains high level of progesterone and high level of estrogen. High level of progesterone and high level of estrogen, it will stop your body from producing luteinizing hormone. So when there's no luteinizing hormone, it means there is no ovulation. You just imagine, can a person that is pregnant get pregnant again when she is pregnant? You get what I mean, right? Okay, not possible, right? The person cannot be pregnant again when she's pregnant, because the body will not produce the secondary oocytes for fertilization of sperm. So you cannot get pregnant when you are pregnant. There are cases like that, but it's very rare. Very, very, very rare. When a person is pregnant, but she's pregnant again when she's pregnant, okay? Very, very rare case. Super rare case, and it's quite dangerous for the mother, and also very dangerous for the fetus as well lah. So that's why contraceptive pill is to stop you from releasing secondary oocytes, which is to stop you from ovulation time. So you can see that contraceptive pill contains estrogen and progesterone. Pituitary gland is less stimulated and secretion of luteinizing hormone is low. Secretion of luteinizing hormone is low. Okay, a bit ugly the word here. Okay, if you don't want to write luteinizing hormone, can you just write short form, which is LH? You cannot just write short form only. You are encouraged to write luteinizing hormone. You can bracket LH. Then you can write LH at the following question. But at least you must write once lah. Luteinizing hormone and the follicle stimulating hormone at least one time in the long sentence before you write the short sentence, like the short form. Okay, next one. A woman decided to consume contraceptive pill to distant a pregnancy. However, she took the pills at irregular times and frequently mistaking the pills whenever she was busy. 2 weeks later, she was shocked after being confirmed pregnant by a doctor. Explain why. Obviously that she skipped her contraceptive pill, right? Like for example, you have a fever, you have chicken pox, you have fever, right? If you stop taking your antibiotics, like today you eat antibiotics, tomorrow you skip, the next day you eat again. You eat at irregular time. So do you think that you can recover faster? Obviously no, right? You cannot recover as fast as you take the full dosage of your antibiotic, right? So same towards this part as well, contraceptive pill. If you skip contraceptive pill, obviously it's not effective anymore. When it is not effective, obviously that you're gonna get pregnant. Okay, but of course she just want to distant a pregnancy, so it's okay that she's pregnant again, because maybe she didn't want to have a short gap between the kids. Okay, so your answer here, you can see ovulation occurs and menstrual cycle and ovulation return back to normal where blood contains progesterone and estrogen. Low level of progesterone and estrogen. And then you can see follicle stimulating hormone which is FSH, is secreted by pituitary gland, which stimulate development of follicle in ovary and stimulate secretion of luteinizing hormone. Okay, so this is the answer. A bit long, but can lah, okay. Can say contraceptive pills inhibit secretion of FSH ma? So for that question, right, you have to talk about the ovulation instead of FSH because the question says N, right, which is ovulation. If you want to write FSH, then it's another question already. Okay, so next question. This is Form 5, Chapter 5. I think that actually this question, right, to some people, they would think it's very easy because they don't need a lot of biological knowledge. Whatever you see, you just fill in the blanks. So if your theory is very bad, maybe you can try more on Form 5, Chapter 5 question by schematic diagram and the Punnett square. You will score a lot of marks by understanding the question itself. I don't know how many of you understand well because I have a lot of students. They will always choose this question to do first because this is like the easiest question in biology. As long as you understand the question, okay, it says 5.1 shows the genetic diagram of cross between a yellow base color and a speckled winged parakeet with white base color and non-speckled winged parakeet. Parakeet is a type of parrot. Big B is the dominant for yellow base and small b is the recessive for white base. Big G is dominant for speckled wing and small g is for recessive for non-speckled wing. So the question they give you, one of the parent is yellow base speckled wing, the other one is white base color non-speckled wing. This question here is very simple. Why? Because they give you the genotype. They say both are homozygous, dominant, and recessive. This one is too big B and too big G, right? So this is homodominant. Homodominant. Okay, and this one here is too small b, too small g. As a recessive, to show a recessive characteristic, it must be small letter. So this one confirm it is recessive. There is no hetero here. What does hetero means? Hetero means two different alphabet. For example, big B, small b, big G, small b, G is called hetero. Heterodominant. Okay, this one is called heterodominant. If you don't know what is hetero and homo. Hetero is different, homo means same. And then you have to fill in for the gametes. So how do you want to find the gametes? If you don't, never mind, they give you a hint over here, right? This one is your hint, right? So you see it's 1B, 1G, right? Same thing. This one is 1 big B, 1 big G. You color properly, okay? The one is on top, one is at the bottom. So this big B, this is big G. Then you have your gametes already. Because at the question they give you this hint, right? You just copy it like from the question itself. Next one, genotype. So how do you want to find your genotype? You just put the B and B together, G and G together. So your genotype, right, supposed to be big B, small b, big G, small g. Remember, dominant in front, recessive at the back. You cannot write big B, big G, small b, small g. No, same character, same alphabet, you must put it together. Different alphabet, you put it together. Dominant at front, recessive at the back. So this is the correct one. Big B, small b, big G, small g. And of course, you're going to draw the diagram, okay? So this is big B, small b, and then this is big G, small g. Okay, you're gonna draw the diagram out. So this is the answer. So it's very simple for at this moment. Next question, they ask you to write, state the phenotype of F1 generation. What is the phenotype? What is genotype? Gene is the alphabet A, B, C, D, E, F, G. It's called genotype. Phenotype, it means the physical characteristic. Whether they are yellow or whether they are white, whether they have speckled or non-speckled wing. since they have dominant, right, which is big B and big G, right, you always follow the dominant characteristic when there is dominant alphabet. So big B refers to what? Big B refers to yellow, right? So is yellow base color, and big G is the speckle wing, right? So this is yellow base color and speckle wing. Okay, speckle wing. So this is the answer. All of the F1 generation, they have the same characteristic. Yellow base color and speckle wing. You can refer to the diagram because they give you a bird that is the same as the parent, right? So you can refer to it to get the answer. State Y cross shown in 5.1 is the dihybrid inheritance. What is the dihybrid? Di means two. Okay. What does two means here? Two, it means they have two traits. Means two character. Over here, what are the character? Number one, they have the base color. Number two, they have type of wing. So this is what we call as a dihybrid. If it's a monohybrid, right? Mono means one. If it's only one, it can either be base color only or it can be type of wing only. That is mono. So remember, mono, there's only one trait, one characteristic. Di means they have two characteristics. Your syllabus, you only have one and two. There is no more than two one. So you can see if it's one, it's mono. If it's two, it means di. It's very easy to differentiate. This one obviously is dihybrid because you have two characteristics, which is base of color and type of wing. So your answer is involve two traits which include base color and type of wing. Base color and type of wings. Okay, next one. Table 5 shows the Punnett square of a self-cross between F1 generation to form F2 generation. Self-cross means what? It means you use F1 times F1. We call it as self-cross. F1 times F1. So just now, F1, what do we get for genotype? We get big B, small b, big G, small g, right? This is the F1 generation, right? And then the other one is same as well. Big B, small b, big G, small g. And then you have to find the gametes. How to find gametes, guys? Very easy. This one, join with this one, you get big B, big G. The first one, join with the fourth one, you get big B, small g. The second one and the third one, small b, big G. And then the last one is small g, small b, small g. You must come from, you must start from B first instead of G, okay? Because they give you color first before they give you the type of wing, right? Then, if the question did not give you any genotype at the first question over here, right? How do you know whether B first or G first? You have to read the question. Because the question says what? A yellow base color, right? It means you're going to start from yellow first, which is the B first. And then the speckle wing is G, right? So you must follow the question. You cannot write G first because the question starts from yellow base and speckle wing. Unless the question says speckle wing and yellow base. So you have to read the question in order for you to find which alphabet comes first. This one, you know, because they give you this answer, right? So this is big B first and then G second. Okay, so we're gonna fill in the answer over here. Oh, hi, Liang Jie, Gao Qiao Liang Jie. Okay, hi, Liang Jie. Okay, so we're gonna fill in the answer over here. So since we have the first one already, this one big B, big G is over here, right? Second one is this one. This one, we have big B, small g. Third one, we have this one, small b, big G. And then last one is small b, small g, right? So same goes to this part as well. You just fill in the blanks. And then next, you have to find the genotype for this one. Hopefully that you guys know what is the genotype over here. How do you want to find? Just put this down and put it over here. Remember, B and B together, G and G together. So your answer for this one is big B, small, okay, I use big color, a bit not sharp color. Big B, small b, big G, small g. Okay, another one over here is this one here and this one here, right? So we have big B, small g, small g. This one is small b, big G. And then this one here is what? Cannot see. Big B, small b, big G, small g. Usually, you don't get it wrongly unless you have careless mistake. That's all only. Usually, people get careless mistake lah. Sometimes when I do a lot of question like that, I also get careless mistake. So you have to make sure that you don't get careless mistake. You have to double check your answer for Punnett square, because sometimes when you get very excited, right, you write very fast, right? Because it's very easy, ma. You write very fast, right? Then you will write the wrong alphabet, like capital letter or small letter. So you have to double check your answer, lah. Okay. Okay. So this is the answer. Very simple. Next one. Not recessive must be at back, like no, okay. The alphabet must be the same sequence. It must be big. It must be B first and then G. You cannot write G and then B. This is the wrong sequence. You must follow all of the same sequence. If it's B first, everything must start from B. Because why this one can be at the front? Why this B can be at the front? Because B and G is two different character. B refers to color, right? G refers to type of speckle wing. So it's two different character. Only in this situation you can put a B at the front. So it's two different things. So you must follow B and then G. You cannot write G first. Wrong. That one is a wrong answer. If you write G first, all of the entire answer wrong already. You will wrong. You will start to get wrong answer from 2 marks over here and the following question. Everything else will be wrong. Okay. So you cannot get the wrong answer. Remember, the alphabet must follow sequence. B or G first, follow the question. And then remember, capital letter at the front, small letter at the back, same character, put it together. Determine the probability of having a yellow base color and non-speckled wing parakeet as F2 generation. For all of the answer over here, whenever you see this question, this type of question, homodominant, homodominant that cross with a recessive, homorecessive, okay, homodominant cross with the homorecessive, your answer, confirm plus chop lah, confirm plus chop for F2 generation, right? It will be 9331. It's a fixed answer, 9331. But of course, you cannot just write 9331. You have to write the how many 9, which is what thing, and then 331. So what are the combination that you will have over here? The combination that you will have, okay, let's base on this diagram here lah. First, this is your first combination. Yellow base color and speckle wing, the first combination. Second combination, you will have this one, white base color and non-speckle wing, second combination. Third combination, you will have this one, yellow speckle color, yellow base color and non-speckle wing. Okay, this one here is the third combination. The fourth combination, it will be white base color and speckle wing. This is the fourth combination. So you must have this fourth combination in your answer. So you have to count by yourself lah. How many 9, 3, 3, 1? 9 is what, then 3 is what, and then 1 is what. So if you count everything, the 9 here refers to, wait, let me use back my red color. The 9 here refers to yellow base. And then they have speckle wing. This one you have to count. Yellow base, speckle wing. And then next one, 3 refers to yellow base and it's comma, non-speckle wing. The another 3 is white color base and speckle wing. The last one is white base and non-speckle wing. Now this one you have to count by yourself, okay? So it's 9 yellow base speckle wing, 3 yellow base non-speckle wing, 3 white base, and then with speckle wing, the last one will be white base and non-speckle wing. So this is the answer for 29331. Confirm is 9331 lah. If your answer got more than 16 right, your answer is wrong. 9331 is 16 right. So your answer must be 16 only because your Punnett square is only 16 boxes. State the phenotypic ratio of F2 generation. So as I mentioned, what is the phenotypic ratio? Phenotypic ratio is basically referring to, oh no, I read the wrong one. Okay, so I read the wrong part already. So this one here is supposed to be here. This is ratio. I read the wrong part. Okay, you bracket the thing, you move down because this is the one I read over top part is the ratio. Wrong part. Okay. By the way, this one is yellow base color and non-speckled wing parakeet. Okay, sorry, this one I write the other way around already. So right now you have to count how many yellow base color and non-speckled wing. So this yellow base color and non-speckled wing is based on the question over here. Yellow base, it must be dominant, right? Which is big B, or big B, small b. This is yellow base color. Non-speckled wing, it must be a recessive. Recessive is too small g, okay? Too small g. So over here, you have to find, in this diagram here, which one here is dominant B and too small g? Dominant B and too small g. So you will have number 1, dominant B and too small g, one here. And then you have another one at this one, dominant B and too small g. Next one, dominant B and too small g. So out of 16 boxes, you get 3. So the probability is 3 over 16. So remember to find the dominant B and too small g. So the answer is 3 over 16. Okay, I just put it the other way around so you can know that it's the other way around. 3 over 16. You can write 3 over 16 or you can write in percentage form, but I think 3 over 16 is the easiest one to get lah. 3 over 16. Next one, determine the ratio of genotype, which is heterozygous and recessive homozygous genotype. So same thing, you have to count heterozygous and recessive homozygous. Heterozygous means what? Heterozygous means it must be big B, small b, big G, small g. Homo recessive, right? It must be 2 small b, 2 small g. This is the ratio that you have to find. You have to find how many 1 big B, 1 small b, 1 big G, 1 small g. So we're going to count this one first. Big B, small b, big G, small g. So number 1, we have it over here. Big B, small b, big G, small g. Next one. Big B, small b, big. Whenever I read this Punnett square, I feel like my life is miserable, okay? So y'all find out, okay? So next one, the third one is this one. This one also big B, small b. Oh yeah, not this one. Big B. Where is it? Wait. I forgot where is it already. Okay. Hectoro 1, 2, 3 over here, and then this one over here. Okay, so this is the answer. So we have 1, 2, 3, 4. So the answer is 4 for heterozygous. 4. And then we have the one potato over here, which is the homo recessive. Small b, small b, small g, small g. So it's like 4 over 1. So the 4 over 1, you have to make sure that you put it into a better way to write it lah, which is you have to write, we have 4 big B, small b, big G, small g. Yeah. And then the 1 will be 1 big, 2 small b, 2 small g. Really, whenever I do this question, whenever I read it out, I feel like I'm wrapping and wrapping again. Okay, so this is the answer. Next one. Based on phenotype of F2, explain how variation occurs. Okay, variation occurs due to 3 things. Number 1, we have crossing over. Crossing over is under prophase 1. Prophase 1. Next one, we will have independent assortment. Independent assortment. Independent assortment is under metaphase 1. Last one, we have random fertilization. So we have 3 ways of getting different type of variation. So you can choose either 2 to explain from here because you have to explain your answer, right? You can choose to write crossing over where they will exchange genetic information at prophase 1. Independent assortment is how they arrange in metaphase. Okay, let's say they arrange like that, it's different. They arrange the other way, is different genetic information. So maybe both of your parents is like good looking, and maybe you are the potato. It can be this reason as well. It can be prophase 1, it can be metaphase 1. So to ensure genetic variation, it's not just prophase 1. It can be metaphase 1 as well. So let's write the answer to explain the answer. Whichever 2 that you want to explain also can. So let me choose independent and random fertilization because prophase 1, we always write the answer, right? Exchange genetic information between 2 non-sister chromatids. So we will practice the other 2 that we don't really write that much. So you can see that during metaphase one independent assortment of chromosome occurs. We have to explain the answer, right? You can say different combination of maternal and paternal chromosome. Next, we can explain about random fertilization occurs where it results in new combination in F2 generation. So this is the answer for variation occurs. Okay, so we are done with all of the subjective question. We're going to try AC question. Section A, we are done already. So section A, we spent about 1 hour, 10 minutes to do section A. So I think you will do faster in school because I explained a lot of things besides just writing, right? Next one, section B, 4 choose 2 question. Of course, we're going to do all 4 questions today. Just in case, whichever question that I didn't discuss is your choice, right? So we're going to do all 4 questions today. I will try to write as short as possible, or I will try to write just keywords only. Unless I have to form sentence, then I will form sentence with you all. So this is Form 4, Chapter 7 under respiration question. It says, diagram 6.1 shows the bark surface on a woody stem. Explain how the adaptation of part in 6.1 provide a surface area for respiratory gases exchange between the plant cell and the environment. So for this question, right, you have to know what is at the hole at the woody stem. In order to carry out gases exchange, the plants can use 2 areas. Number 1 is stomata, which is at the leaf. Number 2 is lenticels at the stem and the root. This is the stem, right? So obviously this is a lenticels. So lenticels arrangement is loosely arranged so that it's easier for gases to move in and move out, which is to carry out gases exchange. If the arrangement is very tight, right, the gases cannot move in and move out so easily. So the rate of diffusion will be lesser as well. So we have to explain the characteristic of the lenticel because adaptation means characteristic. So we have to say that is the lenticels first. So you can see it is lenticels in diagram 6.1. Cells are arranged loosely and has air space. Next one, you can explain about the oxygen and carbon dioxide. So it allows diffusion of oxygen into plant cell and carbon dioxide to diffuse out from cell. These provide a direct exchange of gases. So this is the answer for lenticel. It's only 4 marks out, so you only have to write 4 sentences over here. Okay, Ken, next question. Also Form 4 Chapter 7 under respiration. They give you 2 different types of plants over here. 6.2a and 6.2a and 6.2b shows the Impirata cylindrical and the Oryza subita in a waterlogged paddy field. So it doesn't matter what is the scientific term because they give you a English word over here. They give you a lalang and a paddy plant. So they ask you why these 2 different plants, right? They have different ability of survival in the water. It says what the Oryza subita can survive in waterlogged area better than the Impirata cylindrical. Explain why. Paddy field, okay, talking about immersion into water because this one got water, right? The plant that can survive in the water, we only have two type of plant, which is the one that can survive through anaerobic respiration. We only have 2 types. The first one, we have paddy plant. It's a plant that can survive through anaerobic respiration. The second one is when the plant's undergoing germination. Germinating seed, they can carry out and they can survive through anaerobic respiration. But the rest of the plant, they cannot survive through water. We don't talk about mangrove swamp because mangrove swamp is another type of plant already, right? So talking about normal plant, only paddy plant and the seed that carry out germination, they can carry out and they can survive through anaerobic respiration. Why? What is anaerobic respiration? For those who don't know what is anaerobic respiration, let's write the process out loud, okay? Anaerobic respiration means you partially break down of glucose without oxygen into ethanol, carbon dioxide, and energy. So since you produce ethanol, they will have an enzyme over here. The enzyme we call it as dimase. So they will produce ethanol. So ethanol is toxic towards the plant. So that's why the lalang, the Impera cylindrica plant, it will die because they cannot survive and they cannot tolerate that amount of ethanol. But paddy plant, they can tolerate the amount of ethanol in the water. Why? Actually, paddy plant has a very special characteristic. You don't have to know for KBSM syllabus, but for KSSM, the new syllabus, right? I don't know whether it will come out for SPM or not, okay? Because this one happens in, this one they learn in new syllabus. So paddy plant, it has an enzyme. The enzyme, it is known as alcohol. Alcohol dehydrogenase. Okay, this alcohol dehydrogenase is an enzyme. This enzyme, they can break down ethanol into non-toxic carbon dioxide. So that's why the paddy plant can survive through, because they have this enzyme called alcohol dehydrogenase enzyme to break down ethanol. But the other plant, they don't have the enzyme, right? So that's why they cannot survive through. So in your syllabus, since we don't learn this alcohol dehydrogenase, just in case in the exam they ask you and they give you this example, okay, it's because that paddy plant has a higher tolerance towards ethanol. That is your key sentence in your answer. Higher tolerance towards ethanol, so they can survive through. So we're going to form 6 sentences over here. You have to explain the process, the anaerobic respiration, what do they form, and what happens when the plant exposed to ethanol. So you can say that the paddy plant are adapted to carry out anaerobic respiration, where it can survive even if the amount of oxygen is low. Next, you can mention that the paddy plant carry out incomplete breakdown of glucose. We don't use oxidize because there's no oxygen, so we use breakdown, incomplete breakdown. If it's present with oxygen, we can use oxidize, which is aerobic respiration. So this one we have to use breakdown of glucose. And then you can say it produces ethanol, energy, and carbon dioxide. The cell root of paddy plant able to tolerate high content of ethanol. Paddy plant has fibrous root. So this fibrous root, right, is to enable them to get oxygen on the surface of water, to enable absorption of oxygen from surface of water. So this is the answer. So you get 1, 2, 3, 4, 5, 6. So 6. You can choose to write in paragraph. You can choose to write in key point sentences. Not keyword, guys. When you talk about point form, right, it must be in a sentence, okay? This is like no key. Like dash sentence, dash sentence, dash sentence. You can choose it in paragraph. But I prefer to write it in this way because it's easier to count how many sentences do I have instead of a paragraph. So if it's paragraph, right, then you have to count, oh, this is one sentence, is one sentence. So it's easier for the marker to mark also. But remember, it's not keyword. It's a short sentences when you want to write in point form. This is a point form. It must be in a short sentences. Okay, next question. It's a very popular thing that happens in the youngster, it happens in the old people also. So it's between cigarette and e-cigarette. It says 6.3 shows the usage of cigarette and e-cigarette amongst teenagers that can cause bad effect to health. For 6.4 shows the differences of volume of substances that are present in cigarette and e-cigarette. Compare the substances that are present in cigarette and e-cigarette and the effect to human health. Okay, for this question here, right, it says compare. I have student who asked me, teacher, when do I need to write similarity? When do I need to write differences only? Whenever compare, if you don't know what to write, you write both. You write similarity and differences. You can choose to write one similarity. As long as you have similarity, right, it means that you have to write like more differences, right? If you write two similarity, you're gonna write 8 differences. So it depends on how many similarity that you have, and you can write the remaining one. Of course, you can write more differences lah. So you choose to write similarity and differences both at the same time. Okay, if you do not want to write both at the same time. Okay, so from this question here, you can see that the e-cigarette and cigarette content, okay, based on this table here, you can explain your answer. I know that you guys don't learn e-cigarette in bio syllabus, but you only learn cigarette, right? So if you compare e-cigarette and cigarette based on this table, you will have your answer. First one, you get the similarity first. What is the similarity? Both contains of nicotine that it will cause addiction. Both it has heat that dries up the alveolus. Both have carcinogen that it will cause cancer-causing substances. So you have 3 similarity. Then you're going to write 7 differences, right? So you can compare the content where at e-cigarette, they have more nicotine. And then for cigarette, you have lesser nicotine. And then heat. E-cigarette will have more, e-cigarette will have lesser. Carcinogen, e-cigarette will have more. So you based on all these differences, you form into sentences for differences. Okay, just based on this table. Don't have to base on anything else, just based on the table. But your explanation, of course, you must base on your biology. Like for example, nicotine, you must know is addictional. Like heat, it will dry up the alveolus. So this is the explanation that you have to know. If you do not anything, like for example, let's say you are like really weak in biology, if you don't know anything, right, then just write the comparison without explanation, you also get the marks one. Not a full mark, but you will still get the marks. Even if you just write the statement. Like cigarette got more of nicotine, e-cigarette got more nicotine, cigarette got lesser nicotine, right? You also get 1 mark. But maybe you don't get the explanation mark lah. So you don't leave it blank. Just write something there, okay? But I think this one you're very good at it, right? Since e-cigarette and cigarette, right? I have students who this one you're hopefully that I don't catch you who do this in my class, okay? I will not see you anyways. But yeah, okay. So similarity. For similarity, you can say both contains of nicotine, which cause addiction. Both contains of heat, which dries up bolus, or respiratory tract also can. Both contains of carcinogen, which can cause cancer. Okay, now done with similarity, we're gonna write differences. So differences, you can choose to write in table form, but if you don't want, also doesn't matter. Because I want to write faster, so that's why I write in table form, okay? You can choose to write it in sentences. Just that right now, I want to write it faster. Cigarette. This is one is e-cigarette, one is cigarette. Okay, first, we're going to compare the nicotine amount. So based on the diagram that you can see, this one got more nicotine, right? This one has lesser nicotine. If you're able to write this, you're going to get your first mark, okay? If you're able to write this out, okay? More nicotine, lesser nicotine. So when you have more nicotine, it will cause more addiction. Or basically, you will feel excited, or you will feel you will release more dopamine, so you will feel happy. So that's why people always smoke after they eat a have their meal. There is a Chinese saying which I very hard to convert into English. It say what? 饭后一根烟，快乐像神仙. You know, it's like after you eat a meal, you have a cigarette, you will be very happy like a god, something like that lah. So that's why this the reason why people have five. Okay, I don't smoke, so I don't know, guys. I don't know what's the feeling of it. Hopefully that you guys don't know as well. Okay, second one, we have tar. This one has tar. This one got no tar. Based on the diagram, one got tar, one got no tar, right? Based on the diagram, okay? So everything is based on diagram only. So since we have tar over here, you can explain by saying that tar covers surface of alveoli, or alveolus also can. It reduces gases exchange. Okay, so next one, what do we have? We have heat. So you can compare the amount of heat. Let me draw downwards a little bit. Okay, so third one is heat. So this one got lesser heat. This one got more heat. So since they have lesser heat, this one will release water vapor. So if you notice that people who take e-cigarette, right, they will have a lot of smoke coming out, okay? This one, more heat is because there is combustion that increases temperature. And then you can explain by saying that it reduces moisture of alveolus gases cannot dissolve easily. Okay, gases cannot dissolve easily. Next one, the fourth one, we have carcinogen. Yeah, wait. The fourth one, we have carcinogen. This one got more carcinogen. This one got lesser carcinogen. So if it's more carcinogen, means you have high chances of getting cancer. And then the last one, the fifth one, it will be carbon monoxide. So this one got lesser carbon monoxide. This one got more carbon monoxide. So if you have more carbon monoxide, you will experience shortness of breath. Why? It's because that carbon monoxide will have a higher affinity to bind with hemoglobin to form carbon monohemoglobin. Carboxyhemoglobin, which it will lead you to have lesser oxygen to bind with hemoglobin. So that's why you have difficulty in breathing. People who smokes a lot, okay? So this is the reason why. I'm not saying that e-cigarette is better or cigarette is better. Whatever you do, you just base on the diagram. Like they give you a table, right? You base on the diagram. When you base on the diagram, you explain based on your biological knowledge, you should have your answer correctly already. So this is the cigarette related thing. Okay, now done with this one, we're going to move towards to question 7. Question 7 is Form 5, Chapter 1. So this Form 5, Chapter 1 is about heart question, and we have to explain about heart as well. So based on this question, it says 7.1 shows the internal structure of human heart. X is the structure found in the right heart. Okay, so as you can see over here, it says, but this is explain what will happen to the flow of blood if the structure X is not functioning. Whenever you look at the heart, you have to know that left is right, right is left, right. Right is left, left is right, right. So that's why you can see this is the left side, that one is the right side. We're going to do the basic labeling first. So first of all, we have to know that the blood comes in, goes in, and goes out from here. And then the other one, the blood comes in here, the blood goes out from here, right? You have to know the labeling of the 4 blood vessels from the heart because it's very important. So the one that goes into the heart, which is blue in color, of course, they carry deoxygenated blood. So this is vena cava. Vena cava. The one that diffuses out, which is deoxygenated blood, it will be pulmonary artery because all artery transport oxygenated blood besides pulmonary artery. Next one, we have the one that goes into the heart, which is pulmonary vein. So the one that goes out is aorta. Okay, aorta must have 3 holes on top lah, so it goes to different parts. So this is aorta. And then we have to know the valves in the heart. So there are 3 different valves in the heart. So the first one we have over here, this one we call it as a bicuspid valve. Okay, and then we have this, the one in the middle, these 2 in the middle, we call it as a semilunar valve. The right one, which is the X, it is known as the tricuspid valve. Okay, this is the tricuspid valve. So they only ask you to write what happens when the tricuspid valve is not functioning. If you don't know which one is bi or which one is tri, very simple. Remember Ri and Ri. So the right side is tricuspid valve. The left side, then you will know is bicuspid valve. The one in the middle is called semilunar valve. So since it's tricuspid valve, they are supposed to prevent the blood from going back to vena cava. They are supposed to allow the blood to pump out from the pulmonary artery, right? So that is the function of valve. So if valve X is not functioning right, it means they cannot prevent the backflow of blood. But you cannot just write prevent backflow of blood. You have to explain your answer, right? So you're gonna base on number 1, write the name of X, and you have to say that how do they cannot prevent because it's not functioning. They cannot prevent the backflow of blood. So the blood goes to which part? So the first one you can see X is tricuspid valve. When right atrium contracts, the X does not open, means that the blood cannot go in lah. So the blood do not flow into right ventricle. Okay, or another cases where the right ventricle contracts, X does not close, unable to prevent backflow of blood. So basically, this is how we explain the valve question. Because it's 4 marks, right? You make sure that you have like 4 sentences you can explain. So when one part contract, what happens to the other part? When the other part contract, what happens to another part as well? So this is the valve, okay? Next question is under lymphatic system. So for lymphatic system, right, actually this question came out for many times already. I'm not too sure whether it will come out again or not. But this one, they ask you to compare the two fluids in the lymphatic system. They didn't ask you to explain the formation. They asked you the differences between the fluid. So it says what? It says 7.2 shows a part of a blood circulatory system and the lymphatic system in human body. Compare between fluid S and fluid T. You have to know in lymphatic system we have 3 different parts where the fluid comes from the same place. First part is the blood capillary. In the blood capillary, the fluid, it is known as a blood plasma. Which is S lah, okay? S is blood plasma. Next one, we have the interstitial space. Interstitial space can be any other place. So let me draw like this color, okay? Interstitial space. So in interstitial space, the fluid, it is known as the interstitial fluid, as you can see at the bottom. The last one, we have the lymph capillary. Lymph capillary, the fluid, it is known as lymph, which is fluid T. Okay, so this is the fluid come from. All of the fluid come from the blood plasma. Because of high hydrostatic pressure, the fluid will move into the interstitial space, and due to the pressure as well, the fluid will return back to the bloodstream. But you don't have to know the formation. You just have to know what is the difference between fluid S and fluid T. S and T got what differences? So same thing. If you don't know what to write, whether you have to write similarity or differences, right? You write both. You write similarity and differences. So first of all, we're going to start from similarity first. Similarity is both contains of lymphocytes. What is a lymphocyte? Lymphocytes is white blood cell. In your blood capillary, of course, you will have your white blood cell, right? In your lymph capillary, why do you have your white blood cell? It's because that you have a thing called lymph node. lymph node is the one that contains of lymphocytes. So that's why lymph capillary has high density or high content of lymphocytes compared to blood, but both also contains of lymphocytes. Okay, so you can see the similarity. Both contains of lymphocytes and both contains of respiratory gases. Respiratory gases such as oxygen, carbon dioxide, they will also contain of mineral as well. But respiratory gases is the easiest one to remember because any part of the body you must have oxygen and carbon dioxide, right? So for differences, what is the differences between the S and T? Okay, S is the blood plasma, and then T is the lymph, right? Okay, the differences between S and T, number one is the red blood cell. Blood plasma, it is red in color, right? Why they are red in color is because that contains of red blood cell. Lymph is colorless. Why is colorless? Because there's no color pigment at all. So that's why blood plasma is red, lymph is colorless. So you can see that this one contains of red blood cell. Of course, exam cannot write RBC lah. At least you must write red blood cell. Or you can write erythrocytes. And then lymph got no red blood cell. And you can see this is red in color. This one is colorless. Okay, besides red blood cell, what is the difference? When there is a high hydrostatic pressure, right, they will push all of the fluid to diffuse into the interstitial space. There are three things cannot diffuse into the space: is red blood cell, blood plasma, and platelets. So you can mention about blood plasma has the plasma protein, and then for lymph there is no plasma protein because plasma protein is too large to diffuse across the lymphatic, the interstitial space. So this one you can see this one has large plasma protein. This one got no plasma protein. Okay, so for blood plasma, you can see where can you find the blood plasma, right? So blood plasma can be found in the blood vessel. Found in blood vessel. This one found in lymphatic vessel. Based on the diagram that you can see there. One is blood vessel, one is lymphatic vessel. Okay, last one that we can compare is the lipid soluble substances. So if you compare, right, lymph will have higher lipid soluble substances is because that lymphatic system, they are connected to the lacteal. Lacteal is the one in the small intestine. If you don't remember, it looks something like that. This is your villus. Okay, the one in the middle we call lacteal, right? So lacteal, E-A-L, lacteal, they are connected to the lymphatic vessel. So lymphatic vessel is the one that join with the lymphatic system, which is the lymph. So that's why lacteal is the one that absorbs lipid soluble substances. Hence, lymph will have more fats compared to blood capillary. Because blood capillary absorb lipid soluble, water soluble substances such as water, glucose, amino acid. Vitamin B, C, lacteal is the one that absorb vitamin A, D, E, K, fatty acid, and glycerol. So this one you can see there's lesser lipid soluble substances. Lipid soluble substances. This one got more lipid soluble substances. Okay, so this is the differences between lymph and also blood plasma. Okay, can, because I don't know whether you guys can or not. Okay, can, right? Okay, so next one we're gonna move towards to the next one, which is the hypertension. How do you want to regulate your negative feedback mechanism? So this negative feedback mechanism, okay, it's a very long question. Let me read the question first. It says, hypertension is a serious medical condition that significantly increases the risk of heart, brain, kidney, and other diseases. An estimated 1.13 billion people worldwide have hypertension. Based on research in 2015, 1 of 4 men and 1 of 5 women had hypertension. It occurs when the blood pressure is consistently above 140 per 90 mmHg. Okay, so diagram 7.3 shows the regulation of blood pressure in an individual. The blood pressure is regulated by a negative feedback mechanism. What is a negative feedback mechanism? Basically, it means that you have to regulate back to normal. So let's say right now I have a high sugar level, I have to go back to normal. We call it as negative feedback mechanism. Okay, so let's say for example, right now I have a high blood pressure, right? I have to go back to normal, right? Like back to normal. Also a negative feedback mechanism. As long as you can go back to normal, we call it as negative feedback mechanism. Negative feedback mechanism. So they ask you to explain how blood pressure of an individual is regulated by negative feedback mechanism. It means you have two scenarios. Number one is when the person has high blood pressure. Number two, when the person has lower blood pressure, right? Two, right? So it means that you have five sentence. Five for higher blood pressure, five for lower blood pressure. But the answer is just opposite. If you know how to write the high blood pressure, you will know how to write the low blood pressure because answer is just opposite. Less to more to less, high to low to have that all. That's all only. But first, you have to know at least one thing, which is either high or low, right? So in short, let me write over here first. When a person has high blood pressure, we're going to discuss about high blood pressure. When a person has high blood pressure, your body needs to receive a receptor in order to detect is the one that detects the increasing of your blood pressure, right? The one that detects the increasing of blood pressure, it is known as a baroreceptor. Okay, it is known as a baroreceptor. Speak in Chinese, you cannot understand because my student cannot understand Chinese, so I can only use English, okay? So, baroreceptor, when you have a high blood pressure, right, it is more stimulated. Okay? I already use a very simple English, okay? I mean, if you really don't understand, of course, you can ask me later, okay? So this baroreceptor, since when they are stimulated, they will send impulses to a place called cardiovascular center. This cardiovascular center is in your brain called cardiovascular center. This cardiovascular center, it will send the impulses to your effector. Right now, your effector is the heart muscle. Okay, it is the heart muscle. So since you have a high blood pressure, right, you want to regulate back to normal. Obviously that your heart muscle, it will pump slower. Your heartbeat, it will reduce. Your blood pressure, it will reduce as well, right? So basically, it means they will reduce heartbeat. Yeah, heartbeat. Okay. At the same time, they will have vasodilation. Vasodilation, it can only be used in blood pressure. What is dilation? Dilation means it becomes bigger in size. We call it as dilation. It becomes bigger. It's called dilation. So because right now you have a high blood pressure, right? If your lumen is bigger, it means that the blood can flow more, right? So the pressure is not that high. So that's why we have vasodilation of your blood vessel. So blood vessel is under smooth muscle. So your smooth muscle, relax. Okay. Smooth muscle relax. Next, we will have decrease in resistance of blood flow in blood vessel. Decrease in resistance of blood flow in blood vessel. You have to go back to normal, right? Whenever you talk about negative feedback mechanism, doesn't matter whether it is a blood pressure, doesn't matter whether it's a blood sugar level, whatever, you must write it return back to normal. You cannot say that it decreases or it increases only. You must say it return back to normal. So that's why we have a high blood pressure, right? So you can say blood pressure decreases and return back to normal. Okay, remember to write return back to normal. You cannot just write decreases full stop, because if it decreases, what if they decrease below the normal range, right? Then it's all not normal, right? So you have to say return back to normal. Okay, right now we have to form all of these things here into sentences. Okay, I will try to write one part, and then I think you can try to write the other part as well, which is the lower blood pressure one. So first one we can say is when the blood pressure increases more than normal range when the person has a higher blood pressure, okay? Okay, let's form sentence just in case you all don't know how to form sentence from the keyword just now. First, baroreceptor. We have a few receptor in the body, but we will talk about that later. Are stimulated, it will increase rate of nerve impulses and sent to cardiovascular center, as we did just now. And then next, nerve impulses sent to effector, which is cardiac muscle. Contraction of heart becomes slower, vasodilation, vasodilation of blood vessel occurs. Next, smooth muscle relax. It will decrease in resistance of blood flow in blood vessel. Blood pressure decrease and return back to normal range. Okay, so basically this is what happens when the blood pressure increases. So let's say if you want to explain about when the blood pressure decreases, everything is just opposite. Okay, what do I mean by opposite? It means that, okay, let me use other color so that I don't have to write it again now, okay? If it decreases, right, you have to say baroreceptor are less stimulated. Okay, the one in blue color is to represent when the blood pressure decreases. So it will decrease rate of nerve impulses and sent to cardiovascular center. Lesser impulses sent to effector, which is the cardiac muscle. Contraction of cardiac muscle becomes stronger or faster also can. Stronger or faster also can, okay? Yeah. This one got no less, this one, okay? Just write nerve impulses is enough already. Faster. Vasodilation change to vasoconstriction. Why I cannot write right now? Vasoconstriction of blood vessel occurs, smooth muscle contract. It increase in resistance of blood flow in blood vessel, blood pressure increases and return back to normal. So basically whatever in blue color over here is the one that you have to change when you want to explain when the blood pressure decreases, which you have to write in your exam now because it's 10 marks, right? So the first 5 marks is when the blood pressure increases. The next 5 marks is when the blood pressure decreases. Okay, Ken? So this is the answer that you have to write. So the blood pressure decreases, you all write by yourself now. So I think that it's just opposite, right? You can write by yourself, right? Okay. We're gonna try the next question, which is question 8. Question 8 is Form 4 Chapter 8, which I believe that a lot of students, they don't understand Form 4 Chapter 8. No matter whether it is a mangrove swamp or no matter whether it is not mangrove swamp, they will not understand. But for this Chapter 8 question, right, it is not a K-bar question. It means whatever they come out in this question here is theory based. Theory based means if you study before, means you know. If you don't study before, means you don't know. It's just a normal theory question. There is no K-bar for the question that we are doing right now. It's under Form 4 Chapter 8. Okay, Form 4 Chapter 8. So, this Form 4 Chapter 8, right, you see it's 8.1 shows some of the medicinal product manufactured by the biotechnology. Explain how these microorganisms are used in producing in the medicational products shown in 8.1. Basically, it means that what are the the useful microorganism that we can use it in biotech. Because we talk about microorganism, right? We will think of microorganism, it must be bad. Okay, it must be bad microorganism, right? But some of the microorganism is good. For example, if you want to make vaccine, antibiotic, anti-serum, insulin, everything here they will use a good bacteria or good fungus or good whatever lah. Okay, is to make into a vaccine. Like for example, if you want to do COVID-19, right? COVID-19 is a vaccine that does not contain of any pathogen. They only take the mRNA from the COVID-19 and they make it into a vaccine. So you will not get COVID-19 when you get the vaccination. You only get the mRNA of the virus. You don't get the virus, you get the mRNA of the virus. So don't have to worry about when you are thinking to get vaccine or not vaccine, because you never get the COVID-19 virus. You might have side effect, but you will never get COVID-19. Okay? So we're gonna start one by one: vaccine, antibiotic, anti-serum, and insulin. Everything here is just based on your theory. If you study before, means you know. If you never studied before, means you don't know lah. First of all, we're gonna start from vaccine. Vaccine is produced by a dead organism, a harmless dead organism. So this dead organism, right, it will not cause our body to get the disease. So when we inject the dead microorganism, right, in this case, our body will react to the antigen, the pathogen. Because for every bacteria, for every virus, for every fungus or whatever thing, okay, for everything, they must have a marker. This marker we call it as antigen. Just in case you all don't know what is antigen and pathogen. Pathogen is a harmful microorganism. It means it's the one that causes you to have disease. We call it as harmful microorganism. Antigen, it means that it is a marker. This marker, it means that every harmful microorganism, how would your body know it is a harmful microorganism? Is based on the marker, based on the antigen. So how do I know whether I am me? It's because that I have IC. How do I know it's you? Because you have IC, right? You have fingerprint, right? It's the same concept over here. So let's talk about vaccine. Maybe you have to write like slightly smaller words because page is quite limited lah. Okay, you can just take your whatever paper to write. Okay, vaccine. So first, vaccine. You cannot explain. It says it is prepared from weakened or dead pathogen, or you can say you can say microorganism also can. And you can say it stimulate. Yeah. Cannot write again. Stimulate production of lymphocytes in body that recognize antigen of pathogen. Antigen is the marker. Pathogen is the bacteria of the pathogen as foreign protein. It means it's a thing that you want to remove from your body lah, okay? So what's so special about lymphocytes? Because lymphocytes is a memory cell. It has a memory cell. So they can remember the bacteria that went into the body before. So you can see lymphocytes has memory of same pathogen. If the person exposed to same pathogen in the future, lymphocytes can produce more antibodies to fight against disease because they can recognize the same antigens of the pathogen. It's like no student lah. When you try a question before, you might have a memory of you doing the question before, right? Unless you are like goldfish law. Today you learn, tomorrow you forget, right? Then it's different case, okay? So this is vaccine. Next one, we have antibiotics. Antibiotics, you have to know two of the example of antibiotics. One is called penicillin, the other one is called streptomycin. So this penicillin and streptomycin is a popular question in paper 1. You have to choose which one is the antibiotic. So we're going to talk about these two antibiotics name. One is made from fungus, the other one is made from bacteria. It's the antibiotic that we usually take, so don't worry whether it is a fungus or bacteria. So for antibiotic, so it's a chemical produced by fungus or bacteria, because as I mentioned, right, we have two different antibiotics that we have to know the name of the antibiotic, which kill bacteria. They will bind permanently permanently at cell wall of bacteria when destroying the bacteria. Okay, so this fungus and bacteria, right, is a good one. When you want to kill this bacteria here, is the bad one. Okay, maybe not this color. This color, okay. This one is the bad one. The one in yellow color is the good bacteria. The one that you want to kill the bacteria is the bad one, okay. Now, since I mentioned we have two different types of antibiotics, the first one is known as the penicillin related antibiotic from fungus. The other one is called streptomycin. Mycin from bacteria. These are the two examples of antibiotic that you have to know, because it's a paper 1 question, or even for this essay question, you have to know the example of antibiotics. So next one, we have anti-serum. Anti-serum, it is the one that you inject when you have the disease. For example, you have diarrhea, you have chickenpox, you can inject the anti-serum. How anti-serum is being made? Basically you can make by having injecting the harmful microorganism into the body of the pathogen, or body of the animal. So if you want to get an anti-serum, right, you will inject, let's say you want to get chickenpox anti-serum, so they will inject the chickenpox virus into the horse or into the whichever animal's body, so that the body of the animal will react to the chickenpox virus. So when the animal react to the chickenpox virus, right, the animals will produce a lot of antibodies, right? So for human, at this point, we will take the blood of the animal to make into antibodies, anti-serum. It sounds very scary, but this is how it works. So it was last time lah. Right now we have technology. We can just straight away use the technology to carry out the production of anti-serum. We don't have to use animal because we have animal rats. Bacteria got no rats, so we use animal. This is a Wailing fan cup guy. Hi, okay. So next one, talking about anti-serum. Okay. Some more this time got effort. He put my photo as her photo as the photo. Seriously, okay. So this anti-serum explanation, you can see they are prepared by injecting pathogen into animal's body. The animals will stimulate lymphocytes to produce antibody towards disease. blood serum, or you can say plasma, that contains of yeah, of contains of what? Contains of antibody is extracted to be used in human. Hi, I'm new. Can I use table? You can only use table when there is a differences question. If it's like that, then there's no point for you to draw a table. If you want to draw a table, it must be a different, like compare and contrast question. Only you can write table form. Okay, last one is to produce insulin. Okay, now this production of insulin itself, right? It can be a question itself. It means it can be like 6 marks question instead of like a 10 marks question. It's a 6 marks question. So this production of insulin is under biotechnology, which is genetic engineering, right? I actually prepare a diagram. It's not in your notes, but I prepare a diagram over here so we can use this to describe how does this thing works. Since it's genetic engineering, you need two things. Number one, you need human cell. Number two, you need bacteria. So for bacterial form, right, you can see bacteria is a very special microorganism because it does not contain of nucleus. Okay, number one, bacteria got no nucleus. Okay, no nucleus. So what do they have? They have this thing called plasmid. This round circle thing is known as plasmid. So plasmid is the one that contains of genetic information. It's known as is the one that contains of genetic. Since they have no nucleus, right? So it's the plasmid. So how do they want to take the genetic information out from the plasmid? Is they have to use an enzyme. So yeah, wait. So enzyme is too small. Can I write? Enzymes is used to cut plasmid. Okay, so when they use the enzyme to cut plasmid, is to obtain only specific nitrogenous base. Okay, is to obtain one of the genetic information from the plasmid. Okay, if you want to know the name of the enzyme, it is known as a restriction enzyme. restriction. It's called the restriction enzyme. But if you don't want to write the restriction enzyme, you can just write enzyme also can. So enzyme is used to cut the plasmid, not the scissors, but like a scissors function. They want to cut the plasmid. They only want to take a certain part of the gene from the bacteria, certain part of the gene from the. So they will cut the area, and then what happened next is they will take the human gene. Okay, human gene that contains of insulin. So they will insert into the plasmid. Okay, they will insert into plasmid, which you will see the thing with the red color spot here. So this thing here, it is known as a recombinant DNA. Okay, it's not called plasmid. Recombinant DNA. Recombinant DNA. So they will put it into a culture. So this culture here is for them to carry out a rapid asexual reproduction so they can produce more. So as they produce more, they can produce insulin. So if you are able to explain nucleus, the bacteria, they will obtain the plasmid, and the plasmid is being cut. The enzyme is being used to cut the plasmid and to obtain the genetic information. And then next one, you will take the insulin gene from human and insert into plasmid to form into a recombinant DNA. You grow it into a culture to carry out a asexual reproduction and to produce in a larger amount. You form insulin. Of course, this is just an experiment. In factory wise, right, they don't use this culture base because it's very slow. So what they use is they use a bioreactor. Bioreactor, they can produce like many, like don't know how many liter of insulin instead of you just growing into a culture. So they will use a bioreactor in factory. But in your syllabus, you just have to explain they grow in the culture. Can I write an answer in point form? Point form, it must be in a sentence form. This one, the one that I write over here is a full sentence point form. Your point form must be in a sentence. If you want to paragraph also can, but it must be a full sentence. Cannot just write keyword. Okay, first one, insulin. Insulin. Don't know if I have space to write or not. So we have insulin. So you can see This is genetic engineering. Genetic engineering. So this genetic engineering, right? If they ask you to explain, they ask you to define what is the genetic engineering, you can write genetic engineering is a alteration of genetic materials of an organism to create new combination of gene. This is the definition of genetic engineering. If they ask you to define now, okay, so it's an alteration. If you don't write alteration, you can write change. But alteration is a better word to use of genetic materials of an organism to create a new combination of gene. Okay, so continue to explain what is this insulin. How does this insulin form? So you can see an enzyme. You can write restriction enzyme, but don't want to write also, never mind. Just write enzyme, okay? Make your life easier. Is used to cut the bacteria plasmid. Human gene that contains insulin is cut. Human gene is then inserted into plasmid, which is now known as recombinant DNA. Recombinant DNA. Recombinant DNA is inserted into bacteria, which grows in culture and produce a large amount of insulin. So when you have this recombinant DNA, right, can you see there's this thing called transgenic bacteria? So they will put it back into the transgenic bacteria. It will not be the original bacteria over here. So it will not be this one, okay? Because this one here already died. You take the nucleus out, means the bacteria already died. So you will put it into a new bacteria in order for them to grow bigger in size. Because if you only have the recombinant DNA, right, you cannot grow in a larger amount, right? So you must put it into a living things in order for them to reproduce asexually. So this is how insulin is being formed. So this one, I think you have to know how to explain the answer, because I think this is quite important as well. But if you know the diagram sequence, then you should be able to form into sentences. Remember, bacteria got no nucleus. The one that take it out is called plasmid. So plasmid is being cut by an enzyme is to obtain the genetic information. So the genetic information, the plasmid, it will then combine with the human gene that contains of insulin and inserted into the bacteria. So this recombinant DNA, when they insert into the bacteria, it will left it in the culture medium for them to grow. So when you have more, it means that you can produce larger amount. This is how you produce insulin. Okay, next question is biodiversity is a variety of plants, animal, and microorganism living in the earth. This organism lives in different ecosystem and importance to our life. Discuss the importance for the conservation of biodiversity. This question sounds very complicated, but actually it's very simple. Conservation means you have to protect the biodiversity, means the entire world means the world okay. So we have plants, animal, and microorganism. We're gonna base on this 3 points to explain the answer. So you just write out what is the importance of having animal, plant, or even microorganism. So for example, we have a source of food, and we need to have food, right? Based on these 3 things. We can also write provide shelter, provide buildings, build buildings by having the timber. You can also mention about prevent soil erosion. You can mention about water catchment area also can. So just write all of the advantage of having these three things in the world. Then can already. Okay. So first, you can write source for food source. Okay. Number two, you can say provide timber to build houses. You can talk about maintaining a nitrogen cycle, carbon cycle, or whatever cycle, okay? Because once you have three, then the process can take place. You can say they can prevent flash floods, prevent soil erosion. You can write a for educational purpose. For example, we have the swim, which is a reserve forest. You can go there and study, but you cannot pluck anything from there lah. You can say provide income for certain area by acting as tourism spot. You can also write to maintain stable population to maintain stable population by allowing biological control. Okay, allowing biological. How many points? 1, 2, 3, 4, 5, 6, 7, 8. So we have 2 more points to go. Then you can write. What did I write? I also forgot already. Regulating climate condition means there is no global warming or even greenhouse effect because we have not enough of trees. And then you can say, what else can I write? Source of food, provide timber to build houses, maintaining nitrogen cycle. Wait, let me think of one more point to write. Oh, source of medicine. So traditional medicine, for example, we have what tongkat ali lah, we have what Jat Fatima lah, ginseng lah, those are from the forest, right? So these are the example. So you just write anything. There is good thing about environment, having trees, having animals. That is your answer lah. So usually for this type of answer, the answers are very short. You just find this keyword over here only. Or you can say prevent global warming also can. I forgot to write that one, but you can write prevent global warming. Prevent reduces greenhouse effect also can. Okay, so last question, question 9. Question 9 is air pollution question. So this air pollution question, right, it is quite easy and it's very important as well. So the question over here, it says, can I write what? Tongkat Ali, you can write Tongkat Ali, you can write Kicap Fatima, you can write ginseng, but don't write weird thing, guys, okay? Don't write weird thing, please. Second 9.1 shows the infographic of air pollutants index API that cause air pollution phenomena in Malaysia. So the graph, the API here doesn't matter. Describe how air pollution phenomena occurs. So air pollution, it can be haze, it can be the gases released from the factory, from the vehicles. For example, they will release oxide of nitrogen, they will release sulfur dioxide. So they will form into acid rain. So basically, acid rain is one of the example of air pollution. Haze is also air pollution. So depending on where does the air pollution come from, whether it is from the burning of forests, whether it's from factory, from vehicles, or even from a still air. So when the air is not moving, let's say there's no wind, right? When there is no wind, it means that basically all of the haze, it will stay in the same place. So let's say right now Malaysia got haze, right? You will wish that the God will rain, right? So when it rains, it means that they'll bring away the haze away from the area itself. So that's why you need to have a window in order for you to, prevent that much of still air at the environment. If not, you will feel very suffocated, especially people who have asthma. You will feel like dying during haze time. So you can see the phenomenon. The phenomenon is haze. And then you can see open burning. Open. Why my pen cannot write again? Very good pen. Okay. Open burning produces carbon dioxide, oxide of nitrogen. You can write any other gases if you want to. Carbon monoxide also can, which trapped by particles present in air. Next, a factory. My pen is tired now. My pen don't want me to write anything. Okay, again, okay. Factory. Wait, let me wipe my screen a little bit. Maybe it's my sweat. Okay, so you can see factory. 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 Hello, pen. Why cannot write here? Here can write, but the other part cannot write. Why? Factory releases dust. Dust. Hello, dust. Why? Why my pen like that again? Dust and smoke. So usually the factory releases those dust and smoke is because that they didn't install the catalytic converter. So the catalytic converter is supposed to convert the dangerous gases into a harmless gases, right? So if they didn't install this catalytic converter, eventually the gases will be a dangerous gases. It's all done by human one, because if the factory is they install with the catalytic converter, right, there will be lesser pollution. But in that case, you have more pollution. So you can see this is due to human ignorant, ignorant, ignorance. Okay, or lack of knowledge lah. Okay, some area, right, they want to save money. They don't want to like build all these catalytic converter, which is very common, and it happens a lot of time in Malaysia lah. Okay, so you can see it will be worsened by still air, worsened by dry or still air that caused by hot weather, or you can see when there is no wind. So even when there is no rain, they will also cause the same thing to happen as well. So you can see no rain to wash off the suspended particles in the air. Suspended particles in air. Okay, so this is the answer lah. Okay, next one. It says, explain the effect of this phenomenon towards living organism. Obviously, all of the effects here, it will be a bad effect. Because since when you heard of air pollution or haze is something good to human or even living things, right? Obviously, it's bad. Number one, they will have blur vision. So blur vision, it will increase the rate of accident. Number two, you have difficulty problems, where it means that you have asthma attack, where you have inflammation of your lung as well. So this is the other point towards human. Not only human will have, even for plants, they will also have problem as well, because they say towards living thing, right? Not towards human only. So for plants, the dust particle where blocks at the stomata or blocks at the surface of leaf, right, it will reduce rate of gases exchange, it will reduce rate of photosynthesis. When photosynthesis is lower, basically the production of crop yield will be lesser as well. Production of crop yield is lesser means that the people cannot earn money lah, in a way to say that. So when the crop yield decreases, at the same time, it will also affect the food chain. So the entire food chain will be disrupted as well. So your explanation don't have to be so long, just write 5 points is enough. So you can base on human, you can base on plant. human and plant is the easiest one to explain now, actually. So you can say impair human health, such as asthma or difficulty in breathing. Inflammation, eye itchiness also can. And then it says impair visibility that increases rate of accident. Next, dust covers the surface of leaves that reduces penetration of light, rate of photosynthesis reduces. Another one is where the dust covers block at the stomata would be better. Block at stomata of leaves that reduces gases exchange. So when they reduce gases exchange, it means the rate of respiration decreases. Rate of respiration or even transpiration also can. Transpiration is loss of water, also from the stomata itself. Transpiration reduces. Okay, so you can also write it will disrupt the food chains as well. Okay, Kent? Okay, so next question is about human population graph. I mean, this graph doesn't matter because it's not related to the graph anyways. The question says, the world population is now increasing drastically. To fulfill the needs of the increasing population, areas of the forest land must be developed into housing areas, school, factory, and highways. Give an evaluation on above statement with an impact of environment. So over here, since they give you develop into a housing area, factory, school, and highways, right, it means these are the good effects. With the good effect, you have to write the impact. Usually impact can come in good effect and bad effects. Bad effects is easier to write. Good effects is sometimes quite hard to write good effect, but deforestation has a good effect because this is deforestation, right? So we also going to write bad effect. What are the bad effects of deforestation? Number one, it will lead to soil erosion. What is soil erosion? Basically, when the root is not there, they cannot hold the soil. When they're unable to hold the soil, it's called soil erosion. So soil erosion, it will lead to landslide. When there is rain, they will lead to landslide. So it's called tanah runtuh lah, in a way to say that. When there is increasing of landslide, there's no more tree, right? No more tree means that the tree unable to absorb carbon dioxide. It will lead to increasing of your greenhouse effect. Greenhouse effect, it will lead to global warming. So eventually the entire world becomes very, very hot. So this is the bad effect. Not only that, it will have loss of valuable substances. Just like I mentioned, right? The medicine, the timber, the medication. So these are the bad effects. So you can write a lot of bad effect over here. Of course, lesser tree also means that lesser oxygen produced because human we cannot produce oxygen, right? Only plant can produce oxygen unless in the world someone created a technology that can produce oxygen. I mean, of course they are oxygen tank, right? So unless in the future every one of us we have to wear oxygen tank when we go out, not possible, right? So we have to write about a bad effect. So number one, you can say this is deforestation. Deforestation occurs. It leads to soil erosion, landslide, and also flash flood. Flash flood, it means banjir, flash flood. Open burning leads to air pollution. So just now we talk about air pollution, right? The haze, right? So you can say it will have formation of haze. Formation of haze. A more carbon dioxide trapped in atmosphere. In atmosphere. Atmosphere. Okay, why I cannot write atmosphere? Atmosphere. Next, this leads to greenhouse effect. And then you can write it changes the world climate. Climate means the temperature. Season also changes the world climate. It increases the temperature of the earth. This leads to global warming. Okay, you can also write about next one where you have loss of habitat of flora and fauna. Loss of valuable resources such as timber and medicine. I think we have 10 sentences here. 1, 2, 3, 4, 5, 6, 7. Okay, got enough 10 sentences already, okay? Which is the easiest in biology? Is it ecosystem and endangered ecosystem? Ecosystem and endangered ecosystem is a chapter that it will come out for your exam, for your SPM AC question. So if you're very confident with your chapter 8 and chapter 9 for form 4, you can choose that for AC question. Just now we did 2 questions, right? One is chapter 9, one is chapter 8, right? Sorry, it's the same. I think it's the same question. I forgot already. Okay, so that is a chapter that it will come out for your SPM. So I think it depends on your choice. There's a lot of things to study about chapter 8 and chapter 9, and sometimes it will be a K-bar question. So I'm not too sure how well that you can do for your K-bar question. But if you're okay with it, of course, you can choose chapter 8 and chapter 9. So we are done with MRSM paper 2. I will post paper 1 and paper 3 answer later in the Google Drive as well. I will have another class before your SPM paper, which is on the 15th of March, because your SPM is on the 16th, right? Tuesday, right? SPM paper, right? So I have another one more class for you guys, which is on the 15th. So on the 15th itself, supposedly we are supposed to do Penang paper, but I figure it that Penang paper is a bit easy though. So I will do Malacca paper. Okay, so Malacca paper. So Malacca paper is a little bit better compared to Penang paper. So I will choose to do Malacca paper instead. So I will schedule the meeting, and you can set a reminder as well. It will be after Chinese paper because I student will take Chinese, right? So it will be after Chinese paper. Suppose I think it's about 6 or 6:30 I will start the class. Don't know whether it's 6 or 6:30 because I have to find a time where people go back home, shower already. Okay. So I think it's 6 or 6:30. It's one day before SPM. I will be doing Malacca paper too. So if you are not good with your biology and you need some hope in biology, so please come on 15th of March. Okay. 6:30, 6, something like that. Okay. So any question you can ask me. If no question, then you can ask me like commenting over here lah. Okay. If it's not, then I will see you guys on the 15th of March, Malacca paper 2. Okay, hope that you have learned something from MRSM paper 2. So don't always think that papers are very hard to understand. They are very hard to, to write the answer. As long as your basic is very good, it's not hard. Okay, just go back to your basic. Everything related to your basic is okay, then everything is fine already. Okay, so I will see you guys next week. Good luck for your physics and not physics, chemistry and NX paper, guys. Okay, bye guys. Bye. See you guys. 15th of March. Good luck for MX and chemistry and also Chinese paper for those who take Chinese. Okay, bye guys. Bye. Bye, 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 bye. Bye, 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 bye. Bye bye, bye bye, bye bye, bye bye, bye bye, bye bye.